The overall goal of this procedure is to prepare the abdominal epithelium of Drosophila pupae for immunohistochemistry. This is accomplished by first bilaterally dissecting staged Drosophila pupae and removing the internal organs, such that only the abdominal epithelium, composed of proliferating imaginal histoblast and larval epidermal cells, remain attached to the pupal membrane. The tissue is then fixed, incubated with antibodies, removed from the exterior pupal case and visualized through immunofluorescence microscopy to show the gross morphology of the abdominal epithelium and expression of proteins of interest.
Hi, I'm John Yoder. My lab is at the University of Alabama in the Department of Biological Sciences.
Hi, I'm Wei Wang. I'm a PhD student in Yoder lab.
Today we're going to show you our method for dissection of the Drosophila pupae and immunohistochemistry of the Drosophila pupa abdomen. This method can be used to investigate processes of epithelial morphogenesis and tissue reorganization. We specifically use this technique to investigate the development of sexually dimorphic morphology of the adult Drosophila abdomen.
From a 25 degrees Celsius population vial without adults, identify recently immobilized pupae. Pupae with cuticles that haven't yet begun to tan are considered 0 hours APF. Using a wetted paintbrush, gently remove these pupae and place them on the lid of a humidified chamber. Using the paintbrush and PBS, gently wash away debris from the pupa cases. If necessary, sort the pupae by sex. At 0 hours APF, male gonad primordia are easily seen through the pupal cuticle approximately 2/3 of the way down the body. The female gonad primordia are smaller and not as easily identified. Now, place the pupae on the wetted filter paper in the humidified chamber, replace the lid, and return them to a constant temperature environment. When the animals have reached the developmental time point of interest, proceed with the dissection. Be aware that pupae processed 24 to 32 hours APF are most prone to epithelial cell loss during subsequent processing. Prepare for the dissection by adhering a piece of double-sided tape along one side of a transparent slide. Then, use forceps to gently grasp each pupa between the anterior spiracles and place them on the tape, dorsal side up. Line up a maximum of 10 pupae in this manner or risk working too slowly. In 5 to 10 minutes the pupae will be completely adhered, but before this happens, use a paintbrush to space them out evenly and position them in parallel. Prolonged drying at this stage will diminish their quality, so move quickly. Now use a surgical scalpel to dissect each pupa bilaterally. Make a single rapid cut from the anterior to the posterior end of the pupa. If done properly, the cut will bisect both anterior and posterior spiracle pairs, producing symmetric left and right halves. To loosen the pupae from the tape, use a paintbrush to apply a small amount of PBS to each dissected pupa. Using surgical forceps, grasp one pupa half by its anterior end and immerse it in a PBS-filled rinsing dish on the dissecting microscope stage. While still holding the pupa, use a pipetta to gently wash away the internal tissue by pumping PBS into the abdominal cavity. Avoid applying too much pressure or cells will be lost from the epithelium. Immediately submerge the clean tissue in 400 microliters of room temperature fixation buffer. Clean all of the pupa halves similarly. With practice, dissecting and washing 20 pupa halves should take less than 5 minutes. Allow the pupae to incubate in the fixation buffer at room temperature for an hour, and then rinse them 3 times in 400 microliters of PBS for 5 minutes per wash. To store the samples, first rinse them twice in 100% methanol. Then transfer them to 100% ethanol for storage. These samples can be stored in 100% ethanol at -20 degrees Celsius for up to 3 months without any loss of cells or epitope reactivity. To process stored samples, they must first be re-equilibrated in PBS through an ethanol series. Proceed with the immunohistochemistry by blocking the samples in PBS with 2% BSA for an hour at room temperature and without rocking. Rocking samples greatly increases cell loss from the abdominal epithelium. Then, replace the blocking solution with the primary antibody diluted to the appropriate concentration in PBS. Incubate the samples overnight at 4 degrees Celsius without rocking and in a closed humidified chamber to prevent evaporation. The next day, wash the samples 3 times in PBS without rocking and for 10 minutes per wash. Then, block the samples for an hour without rocking in PBS with 2% BSA. Replace the block solution with room temperature secondary antibody in PBS. Then, incubate the samples in the dark for 3 hours and without rocking. Follow the secondary antibody with 3 washes in PBS for 10 minutes each. If appropriate, counterstain the pupae. For example, nuclei can be stained with a 10-minute incubation in DAPI diluted by PBS. Next, equilibrate the samples in mounting media for a minimum of 30 minutes. To mount the samples, grasp each pupa case anteriorly, avoiding the internal pupal membrane. With a second pair of forceps, gently grasp the internal pupal membrane by the head. Then, gently pull the sample from the pupal case. Transfer the uncased sample to a depression slide with 75 to 100 microliters of mounting media. Several samples can be mounted on a single slide. Now, use a probe and forceps to position the samples uniformly in the depression well. Rotate them so that their lateral surfaces face upward and remove any air bubbles with a probe. Finally, gently lower the cover slip onto the samples without introducing air bubbles. For this preparation, structured illumination microscopy will be comparable to confocal techniques. This is a projection of 50 confocal images with 2.5 microns between slices. The tissue is at 26 hours APF, and the staining shows the expression pattern of wingless protein, engrailed-driven GFP, and the nuclei. The anterior is to the left, and dorsal features are to the top of the screen. This movie demonstrates how the gross morphology of samples is preserved by the procedure, thus allowing 3-dimensional reconstruction of image stacks.
Once mastered, several dozen pupae may be processed and placed in fixation buffer in less than 30 minutes. Additionally, samples prepared by using this protocol are amenable to in situ hybridization, making this a valuable protocol for gene expression studies.
Short description. Morphological scaling has been the subject of intense study in evolutionary biology for nearly a century. This methodology permits estimation of morphological scaling relationships across the entire range of trait and body sizes. This was developed explicitly to allow estimation of the wing-body size scaling relationship in Drosophila. It can be used with trait-specific modification to estimate the scaling relationship for any morphological trait in flies. The diet manipulation can likely be used to produce the full range of trait and body sizes in other fully metamorphic insects. Such applications simply require knowing when the MDWE is achieved and applying the manipulation at that point in ontogeny. Thank you for watching. Subscribe if it is useful.